Hi, my name is James Teague. I'm a PhD student at the University of Utah's Robotics Center in the BioRobotics lab. Thank you for your interest in our work entitled Simulating Tendon Shortening During Flexor Tendon Repair Surgery Using a Biomechanical Model and Robotic Testbed. This work is motivated by reconstructive hand tendon surgeries. The goal of these surgeries is to restore functionality of the hand lost due to trauma or neurological issues. There are a wide variety of reconstructive hand surgeries that include different tendon transfer surgeries and tendon repair surgeries. Historically, these surgeries have been developed through clinical and surgical experience. There's no better evidence for developing these surgeries than to examine years of clinical impression, but unfortunately this requires some educated trial and error. With any surgery, the outcomes are a function of mechanical changes of the surgery and the variability of recovery and rehabilitation. Cadavers can be used to investigate the direct mechanical effects of surgery, but they are cumbersome and have limited demographics. While these two methods are the most common for surgical development, new technologies could allow for further investigations. Biomechanical models have been used for a variety of human applications. They provide a way to easily investigate the many aspects of human biomechanics. However, human hand models can require parameters that have little quantification in literature. These models are also complex to use, especially for a surgeon. By having a physical simulator, surgeons could utilize their physical intuition when making decisions. Currently, biomimetic robotic hands are becoming a new tool to investigate different aspects of human hand functionality, including neuromuscular control and biological design. Like cadavers in surgical experience, these robotic hands could leverage the surgeon's physical intuition without the cumbersome cadaver experiments or the potential failures in actual surgeries. Unfortunately, these robots cannot emulate some of the higher performance aspects like tendon and joint friction that a model could if parameterized correctly. While models and robots cannot completely replace surgical and cadaver testing, they could provide a first-line platform for investigating new surgeries or evaluating old ones in new ways before entering the operating room or cadaver lab. To realize this potential, we have chosen to explore a simple but common reconstructive hand surgery using a new biomechanical model of the human index finger and a biomimetic robotic finger testbed. This surgery will be the flexor tendon repair surgery, specifically for the FTP or long flexor tendon. This surgery is performed in response to severe trauma of the flexor tendon, either through tendon laceration or tendon avulsion. Tendon avulsion occurs when the tendon disinserts from the bone. This can happen with elite rock climbers when extraordinary forces are placed on the flexors during holds. When trauma occurs, the tendon is typically repaired by suturing two ends together and splitting the fingers to allow recovery to occur. Tendon repair occurs at an estimated rate of 33 cases per 100,000 people per year, where the FDP of the index finger makes up about half of those cases. Unfortunately, with any surgery, there are plenty of complications that can occur. These complications can occur before surgery, during surgery, or during recovery. The outcomes of these repair surgeries are then evaluated based off of range of motion, grip strength, and presence of functional deformities. Many complications shorten the tendon, which directly affects the outcome of the surgery. However, experimental study of this effect is almost non-existent. Typically, shortening is limited to 1 centimeter. This recommendation is primarily based on the work of a surgeon's clinical impressions during World War II, with only 2 more recent experimental cadaver studies. One looked at tendon shortening's effect on passive extension range of motion and functional deformities. The other examined the effect of shortening on the required forces to make a fist. Both of these studies are limited by issues with cadaver experimentation. Both studies required extremely large forces to move the fingers of the cadaver hand, suggesting tendon adhesion or increased friction. The lack of investigation into this surgery demonstrates the need for new tools for investigating human hand tendon systems. Using a newly developed index finger model, we attempt to do just that. While models like OpenSim have become invaluable tools for biomechanic investigations, many common models severely simplify the tendon system of the human hand. Our new model includes the intricacies of the extensor mechanism of the finger that are almost always neglected. The extensor mechanism is treated like a series of interconnected springs. This model then combines the extensor mechanism with joint dynamics and traditional heel-type muscle models, resulting in the most complete model to date. Alongside our biomechanical model, we have also designed a new robotic finger, Utah's anatomically correct robotic testbed finger, or UART, to emulate the mechanisms of the human finger. This new robotic finger combines the tendon system similar to the ACT hand with the sliding socket joint structures being explored by other researchers. The sliding surface joints can now handle the higher muscle forces required for this type of experimentation, while also providing joint limits in the form of ligaments, emulating their biological counterparts. This also allows for the introduction of movable pulleys required for the proper flexor tendon routing. The robot is driven by DC motors utilizing force feedback from 4 sensors in the tendon routing system. The motion of the finger is measured using a Vicon motion capture system, and the muscle position is measured using motor encoders. While the motors will emulate the muscle forces using hill-type muscle models, the finger's tendon routing and subsequent tendon moment arms become the most important parameter in order to mimic the human hand tendon system. Using a combination of experimental measurement and geometric relationships, the moment arms have been characterized for the robot's tendons as shown in solid lines. While it is known that the moment arms of the tendons vary throughout range of motion, current literature values are limited to the average cadaver moment arms shown in dashed lines. By emulating these moment arms in both the model and testbed, we can now use each platform to investigate tendon shortening during flexor tendon surgery. Simulating the resulting extension range of motion that results from shortening of the tendon is done in 4 steps. First, the hand is placed in a relaxed pose with a shortened but not repaired tendon. Then the muscle is stretched to attach the 2 severed ends of the tendon together, causing an imbalance of muscle forces. Both the model and testbed are then allowed to reach a new equilibrium pose. Finally, evaluating the resulting active range of motion is done by activating the extensor muscles to their fullest as defined by the Hill muscle model. Depending on the amount of shortening, this primarily results in a clod pose with hyperextension at the MCP joint and flexion at the DIP and PIP joints. This simulation was done in both the model and on the robot for either a single or a dual extensor activation over the range of shortening. Single activation shows agreement between the model and the testbed. The dual activation scheme has larger disagreement. We believe the discrepancies are primarily due to the large tendon forces and subsequent large joint friction forces that occur when activating both extensors. Both activations show a deterioration around 10 millimeters, which matches with current clinical recommendations for shortening limits. This video shows a side-by-side comparison of the testbed and model at different levels of shortening. The forces within the Hill muscle model are shown on the right for reference. The largest difference between the model and the robot occur at the determination of the new rest equilibrium position after shortening. This is due to static friction in the robot that is currently not included in the model. Because the muscle forces are small at this stage, they do not cause motion of the testbed. They, however, do reach a similar activation extension limit with severe extension loss at larger shortenings. By using both the model and the robot, we are able to see quantitative justification for clinical recommendations that exist, demonstrating their potential to simulate other aspects of hand tendon reconstructive surgery. In this work, we have developed a new method for exploring the human index finger system using biomechanical models and biomimetic robots with a specific application of flexor tendon repair surgery. Results demonstrate the potential use of these 2 systems as surgical planning tools for future and understudied surgeries. Results also demonstrate the complementary relationship between the robot and model. The robot is physically intuitive to work with, but investigation and emulation of biological parameters is limited for characteristics like friction and stiffness. On the other hand, the model parameters can easily be adjusted to biological values, but this requires physical validation that is lacking in biomechanical literature. Primary limitations of this work are the anatomical accuracy of the model and testbed, and that we are assuming single-digit versus whole-hand mechanics. We are also neglecting the intrinsic muscle contributions and assuming muscle limitations to be governed by isometric Hill muscle models. Beyond this work, and more currently, we are applying this methodology to cadaver experimentation to collect more biologically accurate data. We are also investigating some of the missing biomechanical parameters of the human hand system, like passive joint properties, unmodeled soft tissues, and human moment arm variability. We are also in the middle of expanding and redesigning the UART testbed to include other digits and muscles of the hand, as well as new discoveries we find during the cadaver experimentation. Thank you again for your interest in our work. This work was funded by the NIH's National Institute of Arthritis and Musculoskeletal and Skin Diseases. I would like to thank my collaborators and advisors for their assistance in this work. If you are interested in chatting about the human hand or other parts of my research, please visit me during the poster session or by email. Thanks.